Welcome to our last topic in Chapter 6, which is DNA recombination. In this topic, we're going to talk about the different ways that DNA can be recombined to create some genetic variation. So this topic will talk about double-stranded breaks, which is a little bit of a carryover from our last topic where we were talking about DNA repair. Then we're also going to talk about mobile genetic elements and viruses. Here are our objectives for this chapter, or for this topic rather. I expect you to have these mastered by the time we move on, but I also understand that these may all be brand new concepts to you. So please let me know if there's if you have any problems with these. I expect actually these topic objectives to maybe be the hardest out of all the Chapter 6 topics, or all the Chapter 6 objectives. So the first thing we're going to talk about is double-stranded breaks. Now, we talk about errors in single nucleotide sequences or changes with like deamination, depurination, things like that in the last topic. That's not the only way we can lose genetic information. We can also use this with a double helix break, and where both strands of the double helix are broken. And so that's where we have these two different repair options, which is non-homologous end joining and homologous recombination. Now, the biggest problem with this is that this can result in a huge gap of genetic information potentially. So that's why it's really important that we understand how this works. And before we move on, we need to do a vocab check, just to make sure you're understanding homologous. The definition of homologous is organs or tissues that are similar because of their common origin. So when we talk about homologous combination, or recombination, or anything like this is where we're talking about chromosomes that may not have the exact same genetic sequence, but are very similar in genetic information. So it's the difference between chromosome 1A and chromosome 1B. They're homologous because they're similar, but they're not identical necessarily. So just make sure you understand what we're talking about when we get to that. Alright, so the first one we're going to talk about is non-homologous end joining. So in this case, we get our accidental double-stranded break, and this is the down and dirty method to fixing things. And this can result in a lot of loss of fidelity within the genome. So it's important to understand that this one is the one that will cause a significant amount of loss of DNA. And this may actually explain why we have a lot of junk DNA within our cells, or within our DNA or genome. It's because over the years and decades and generations, etc., we've had these happen, and genes that maybe used to once be functional are no longer functional because we've lost a section of the information. So what happens when this happens is that the nucleases will actually cut on both sides of the double-stranded break and then just sew the chromosome back together, or the DNA back together. So we lose a chunk on either side, if you can see how that works. So there's always a loss in this place, and it's a down and dirty way of fixing things. The way to ensure that we maintain fidelity to an extent is the homologous recombination. And the reason why I say to an extent is because we use the other chromosome to help repair it. So if you, for instance, had one chromosome that coded for brown eyes and one chromosome that coded for blue eyes, and you had this break right at that gene, and it copied from the blue eyes, you might now have the chromosomes that are to pass on for both blue eyes instead of one brown and one blue. And so that's where this will happen. What happens here? So as you can see, the double-stranded break occurs. Then the nuclease will digest the 5' end on either side of the of the double-stranded break. And then what happens is the chromosome will actually do a crossover where it enters into the other chromosome's DNA sequence at the same point of the gene, and will copy the information and then elongate that and have it sewn up by a ligase to create the new double-stranded molecule or DNA. And as I said, it maintains fidelity to an extent because of the fact that there is the same type of code, but the code may vary depending on amino acid sequences. But because of the fact that you had a functional gene before, chances are that you would maintain that functionality. It just might be a different phenotype now. And chances are you wouldn't actually see this expressed in your body necessarily, but it could be something that's passed on potentially depending on where this mutation occurred or where these recombinations occurred. So just be aware that just because if this happened, you wouldn't wake up one day and your eyes would be a different color. So we've seen this happen before, and we've talked about this in previous classes, and this is where crossing over occurs in meiosis. This is exactly how we share those genes, and it's through the same type of process as homologous recombination. And so as you can see, they overlap and they swap each other out. And that's how it works. Now we think of crossing over, every time you see a picture, it's like this whole big chunk of the chromosome will be switched out. And that's not necessarily the case. It's little pieces here and there that get changed out for the other molecules. So it's important to understand that it's not the entire thing. And so that the nucleus will actually break that DNA strand and then reanneal the different parts together, rather than before we were just copying from each other. So make sure you understand the difference between how this works in crossing over versus repairing a double-stranded break. So our next topic, we're going to talk about our mobile genetic elements. These are parts of our DNA that can actually move around within our genome. And so we're going to talk about these a little bit. We're going to talk about what they are, what they do, and we're going to talk about a couple examples. So what is a mobile genetic element? These are called jumping genes. And we have up to 50% of our genome can move around. And that's just phenomenal to me. And I have a hard time even understanding how that's just possible that we can function with 50% of our genome potentially on the move. And these jumping genes can either be introduced by a virus or some kind of infectious agent, or it can be spread cell to cell, or sometimes it can be limited within the genome of that one cell. So you can have a section of chromosome number 1 jump onto chromosome number 2, for example, and that's a really broad example, but it highlights what we're talking about. So it's important to understand that. Now, it's not just bad. There are some good examples where this can create immunity. This is also how antibiotic resistance occurs between bacterial cells. So we're going to look at the human transposable sequences, and there's DNA only, and then we're going to look at some retrotransposons as well. And so we're going to look at both of these options here in the next couple slides. So the first of them are DNA-only transposons. These ones require DNA to move around, and they usually they have to be double-stranded DNA, and they will move from DNA to DNA. And in this case, you can see the two examples we have. We have the cut and paste non-replicative transposition. This is where you will take the transposon, will remove itself from the original DNA and stick itself in the new sequence. Now what can happen here is we can have if we use non-homologous end joining, we will lose a segment of DNA potentially. Or if we use the homologous copy, and then we'll be able to get that DNA sequence, and we maybe won't lose as much information on the original DNA. And it just depends on how we're going to do that. Whereas replicative transposition is where we actually where the gene will copy itself and then insert it. So both the old and the new DNA now have that sequence of DNA. Now this is important because not only is it where we've lost this jumping gene that can be a problem, but it's where this jumping gene inserts itself. If it inserts itself into the middle of a specific gene, that now it could cause a frame shift change or even just nonsense coding, this could cause a significant problem for the cell. So just remember, it could be a silent mutation where it just jumps without a problem, or it could cause a big problem for the cell, just like any other mutation we've talked about. Then we have retrotransposons. And what retrotransposons do is they actually have, they form an RNA sequence off of the retrotransposon sequence that's within the DNA. This then uses an enzyme called reverse transcriptase to read that RNA and create DNA copy that's then inserted into the target DNA. So it's important to understand how that works. And so we use an RNA polymerase that's present in the host to make this new DNA, but it's important to understand that sometimes they'll come with their own information to create this. Sometimes they will rely on the cell's mechanisms for this. So it's important to understand the differences between that. But the retrotransposon will use an RNA intermediate to jump into the other DNA. So here's an example of retrotransposons. The L1 element is our first one, and the L1 element makes up about 50% of our genome. Some of these L1 elements can move, some of them can't, and it just depends. Some of them get frozen into place when they jump. It makes different changes that locks them kind of into place. And this can be a random jump, and actually, this is the jumping gene that's responsible for the hemophilia outbreak we saw in the royal lines of Europe in the 1900s, and that's because Queen Victoria had an L1 transposon jump that was passed to her children and grandchildren that caused hemophilia. And so you can see an example here of how the L1 element moves. So you have the L1 element in the original DNA sequence, makes the RNA. The RNA is then synthesized from the reverse transcriptase and the endonuclease that will then cleave the first strand of DNA sequence, and then it's going to go ahead and use the DNA primed reverse transcriptase to make the new copy of that L1 and insert it entirely into the new DNA. So you can see how this process is happening. If this is a little fuzzy for you, let me know so we can make sure we talk through this together. Another example of retrotransposons are the ALU elements, and these have been implicated in cancer and diabetes and hemophilia. The thing about ALU is that this element does not code for its own reverse transcriptase, so it is entirely reliant on the cell's enzymes to help it move, whereas the L1 will come with its own sequence for reverse transcriptase. And actually what we can do with the ALU elements is that we can compare genetic similarities. So we can look and see if people have ALUs in specific genes and compare how genetically similar they are. And that's actually something that you should be having, or you should have on your schedule for the end of lab this semester, is to be able to do perform PCR on your DNA to find out if you have an ALU element at a specific location on chromosome 16. So it's a good opportunity to just see if you do or not, and that's one of the ways that we can compare lineages is through these ALU elements. So let's wrap up with viruses. First, I'm going to do a little bit of an overview for you on viruses, and then we're going to talk about retroviruses. And viruses are important because they can impact us and impart a lot of DNA and sequences into our cells, as well as the fact that they cause a lot of diseases for us. So it's important that we understand the basics of them. Viruses are pretty interesting little guys. For starters, they're not entirely alive. They are basically a protein ball that contains nucleic acids. Now they can contain either an RNA or a DNA, and those RNAs can be single-stranded, and the DNA can also be single-stranded, or there can be double-stranded DNA. So they have a variety of genome types. They have a variety of protein codes. They have, as you can see here from the image on this slide, they come in a lot of shapes and sizes, and they're very, very diverse. And this is not the virus chapter. We're just going to talk a little bit about the overview here. So you can see in the slide here how the DNA in the virus will enter into the cell and will actually take over the host cell's mechanisms to create more viruses. And so this is how some of these genetic variations can be introduced to our cells. One of the best examples of this is retroviruses. They are unique to only infecting eukaryotes. And they are very similar to how we talked about the retrotransposons, where we have the retrovirus and they carry with them their reverse transcriptase. And what that will do is it will take that RNA and turn it into DNA, which can then be integrated into the host DNA. Now at this point in time, it can either become a latent virus or it can go into active transcription. Once it's been taken over the host cell's mechanisms, then it will go ahead and create the DNA of the RNA and all the protein code needed for continued assembly and replication of this virus. So it encodes its information straight into the host DNA, which is not always the case with other viruses. Other viruses may just take over the machinery and then kill the cell, whereas in this case it becomes a part of that host genome. So it's important to understand how that works. And this is actually partially why we have such a hard time developing vaccines for these kind of viruses. But this is not really the place or time to talk about that. So let's just wrap it up with that so you have the understanding. So this is the end of chapter 6. We went through a lot of different concepts. Please let me know if you have any questions about any of this, and we'll talk some more about it in class.